Riccardo, Jodi, Claire. Hi, thank you very much for joining us.
Hiya.
Hello.
We'll do some introductions first. I'm gonna go to Jodi first. Jodi, why don't you introduce yourself?
Sure, yeah. So my name is Jodi. I'm a postdoc at the moment at the University of Glasgow. I work alongside Terry Quinn, who I believe is in your next session.
Is he?
Yeah.
Oh, you're
I
right.
think so. Yeah.
He is. He's coming up next.
Yeah, so my background's in neuroscience and psychology. And then I did my PhD in looking at brain blood flow and metabolism in healthy individuals so we could understand a bit more about people with dementia and related conditions. And currently I work on a metric called the Brain Health Index. So what that involves is essentially combining different types of MRI in the brain. So what we often do is we will look at one type of MRI that does one thing, but we know, for example, that there's structural change in dementia, but there's also vascular change and there might be hints of small vessel disease. So we have these scans that can allow for us to look at all those things. So why don't we combine those scans and bring a single metric out of it, which is what the Brain Health Index is. So in short, that's what I work on just now.
I'm going to come back to you to give a fuller description of that. Riccardo.
Hello, everybody. I am a research fellow. I am based at the Department of Life Sciences at Brunel University, London. As well, my background is mainly in psychology and neuroscience. I've done my PhD in Sheffield, and I work with MRI, but not only, exactly. So I'm interested in dementia, in aging, and I look at different topics more related to the so-called neuropsychiatric symptoms. But also, I like to investigate a lot of different topics, more biological, but also social. So I guess I will have time to talk about it in details probably later on about those.
Thanks, Riccardo. Claire, did you join us last time? I can't remember.
I don't think I did actually.
No?
I don't know why, but I didn't. Busy. So yeah, I'm a research fellow, an Alzheimer's Society research fellow at the University of Sussex. I'm also a psychologist by background. I'm most interested in kind of cognition and how we can kind of develop tasks which help us identify and understand people who are at risk of Alzheimer's quite early in the lifespan. And so a lot of my work is around like ApoE, E4 carriers, and also some kind of digital health work. But I use MRI now as well.
In that case, I'm going to stick with you because, of course, for anybody who's watching, I don't know if you all managed to tune into the last session. We've just been talking about omics and talking about how there's a good chance that we can probably predict this using genetics and the whole omics spread much earlier on than we do now. And you're mentioning ApoE there. So, Claire, why don't you tell us more about your work, and also if you could add into that what ApoE, what's this all about with ApoE?
Yeah, so ApoE is a really interesting gene. So we all carry 2 copies of the ApoE gene, and there's 3 different variants. So the E2, the E3, and the E4. The E4 variant is the one that's associated with a higher risk of Alzheimer's. And if you carry kind of 2 copies, you can be up to kind of 9 times more likely to develop Alzheimer's than somebody who carries the population norm. So it's super kind of well linked to Alzheimer's. And it's also really prevalent. So about 15 to 20% of people carry an E4 version. So I'm super interested in it because you can see cognitive differences and also structural brain differences, functional brain differences, all the way from early adulthood. And if you can kind of see these differences going on kind of right in the kind of start of the lifespan, surely there must be kind of ways to try and kind of mitigate the impact of this gene from kind of early on.
So you've exclusively worked with people that have ApoE4?
Mm-hmm.
And then is that with individuals, with people? The group that were with us before has really worked with data and data sets. Are you working with individual people?
Yeah, so I work with healthy people mainly, so young and middle-aged, and I recruit kind of a large kind of group of people and then kind of split them into the different ApoE groups. So it's not just E4 carriers, we also have like an E3 control group. And I look at the E2 carriers as well, but not so much.
And so what do you do with these? You know, is this something that, so we're talking about a group of people that have the ApoE gene that we know makes them the at-risk group,
Mm-hmm.
and then you do MRIs on them and give them lots of tests, and how long do you follow them for as well, and at what age do you kind of find those people?
So at the moment, well, like I guess since I started my PhD, I've mainly been working with people aged 45 to 65 as like my key age group, and because that's when a lot of different kind of risk factors start to come online for Alzheimer's, more kind of health related. I don't really do kind of longitudinal follow-up. My current study is kind of longitudinal in nature, I guess, because I'm kind of following people for 8 weeks, but that's something a bit different, which I can chat about if interesting.
Well, no. So that's it. So what's the kind of the aim, if you like? What's the question that you're trying to answer through that group?
So my current project, which is the one I'm looking at with the MRI, so we've noticed that people in the prodromal stages of Alzheimer's disease show patterns of increased brain activity, which we proxy using fMRI. And animal research has shown that these kind of patterns of increased brain activity might actually play a causal role in kind of driving the spread of amyloid and the propagation of tau. So it's kind of a really interesting biomarker. And then kind of what I kind of spotted, and other people did too, is that E4 carriers show similar patterns of increased brain activity in the same regions and networks all the way from early adulthood. I
see. So what you might discover is that there are certain tests or changes that you can find you can apply to people earlier on.
Yeah. So we're trying to kind of understand what the cognitive consequences of these kind of increased patterns of brain activity are. And also whether we can turn down kind of aberrant brain activity using an anti-epileptic in very low doses. And then kind of seeing what the change in kind of brain activity would be following that manipulation.
Oh, so have you done that? Have you tried that yet? You've given people so you found certain people increased brain activity, you give them the anti-epileptic, and then look at them again afterwards. And then I'm guessing though that that's a hypothesis. I mean, you don't know that reducing that brain activity is going to delay onset or change the direction of the disease. Yeah,
it's very much a hypothesis at the moment. We're just over halfway through the study I'm running at the moment. Hopefully it should be done by kind of March, April, so we should have exciting results then.
Cool.
But yeah, it's just a hypothesis at the moment, but fingers crossed.
But again, a great example that we're going to talk about later today is that we've talked this before about teaching old drugs new tricks, is that there might be some benefit to be had from just repurposing what we have. Jodi, tell us more about your work.
Sure, yeah. So as I sort of briefly mentioned, we know that there are like different aspects of the health of the brain that are affected earlier on in dementia. So particularly we know structure is affected, and we know that our vessels can be affected. So we have a variety of different types of MRI scan that all measure slightly different elements, I guess, within the field of sort of structural MRI, that all give us sort of important information. But often when you decide what sort of scans you're going to do, you might pick one or the other. And we have sort of up to 4 different scans that we can use in combination. So what we do is we kind of register them all to a sort of a generic brain so that they're all sat on top of one another, essentially. And then, so on this, we're matching up voxels, which is like if you think of a pixel on an image, and if you had 4 images that were different colors, but you wanted to stack them so the pixels met up with each other, that's kind of what we're doing, but with a voxel which is in 3 dimensions. So then we do a fair bit of complicated maths, but we essentially give each of these pixels a score between 0 and 1 of how healthy they are, and then we average that across the number of scans that we have. and it creates like a mask of the health of the brain, and then we average that mask and we get effectively a percentage score of how healthy the brain is.
And again, I guess the same question I asked Claire, are you working with like random scans that you've managed to get from the hospital, or are you working with individuals who come and get these different types of scans?
Sure. So there's 3 arms to the study. So the first arm was done with UK Biobank. So this technique originated from someone called David Dickey, and I sort of inherited the project from him. So what we did in Biobank was we were essentially trying to find normative values for this metric across sort of an older age range. And then within EPAD, so the European Prevention of Alzheimer's Disease Consortium, they have really good longitudinal data. So what I'm working through at the moment is sort of looking to see how it varies alongside different cognitive scores and over time, over repeat visits. And then there's also the arm which works with the Safe Haven, which is the NHS. It's essentially repurposing NHS scans, so we can then do similar to what we've done in EPAD, but see how it works in the field, because obviously, well, it might not be obvious, in research contexts, we have people who are very, very specialised at MRI who can focus and ensure that a scan is sort of perfectly acquired. So the data is often a lot nicer, whereas in the NHS, we have a lot of people, a lot of throughput, and a lot of people who have to focus on different types of scanning and stuff. And they might have also been scanned for an unrelated reason. So that's a really complicated data set to do it in. So that's kind of the really exciting.
See what I mean? So what do you ultimately, so this brain health index that this informs, or I'm just trying to think that, does this allow, how will that practically come into, is this something that we can practically then put to use?
So it's a really good question, because essentially it's an interesting possible prognostic marker, but then we hit on this question, which I think overarches all of this, the question of, if you could know, would you want to know? And that's the crux of what we face. But from a research perspective, it will enable us to sort of see how other sort of more commonly known risk factors might be playing into it. So, for example, what we're looking at in Biobank is being a diagnosis of type 2 diabetes and hypertension, particularly within our brain healthy individuals. And then also things like BMI, pulse pressure, all those sort of surrogates almost for the concept, well, often surrogates for the concept of obesity and related health conditions, and seeing how they relate to the brain health index. And then,
it's fascinating. Thank you. Thanks, Jodi. I've got more questions about MRIs and things, which I'm going to come back to, and I think you're going to be really good at answering. Riccardo, though, sorry for keeping you waiting. Tell us all about your work, because you've been very patient.
Oh, no, it's okay. Thank you. Well, at the moment, I'm not working a lot on MRI, because I'm working on the project that has been funded by the American Alzheimer's Association. And it's actually a project to investigate cognitive and also brain aging at some point in an understudied population that is basically older people that are from sexual minorities, so non-heterosexual people, that actually have been quite neglected in this type of field. I guess there is a lot of literature about mental health problems in LGBT people, but older people haven't really been investigated much. So luckily, I've got the funding for this project that I thought about 3 years ago. Then there has been the pandemic, and finally I'm starting now to work on it. And of course, it's a complex project, and I will start first with large databases, a bit like Jodi, of data that have already been collected. And at the moment, I'm more looking into cognitive, let's say, comparing different cognitive tests using the English longitudinal study of aging database. So it's a UK-based database to compare these two populations, to have a look for the first time also whether discrimination is actually playing a role in potentially causing, let's say, cognitive decline in these people, even though I'm looking at healthy people, so I've excluded people with dementia or other neurodegenerative conditions. And this would be the first stage, and it would be the first time that I actually, in a study, we will test an hypothesis that had been proposed before but never been tested. The fact that indeed discrimination might play a role not only in causing mental health issues, but also potentially causing cognitive decline in older people. And as a second stage, I will also look at brain imaging using the UK Biobank as well. I guess it's this very large database in which we have a lot of clinical data, brain imaging data. And of course, what I would like to do is always to compare these two populations, but also trying to see whether different risk factors that have already been kind of to some extent tested in this population might indeed explain whether older people might experience, greater cognitive and brain decline. And this hopefully will take me to the last stage of my project, that will be actually recruiting a more targeted sample. Because I guess the drawback of using these large databases is that most of the time they don't include the variables that you want to actually test. So a lot of social variables like personal histories of people, whether they've been discriminated, whether they've had stressful experiences in their life, that are all of the type of risk factors that have been proposed for this population usually are not present in these databases. So finally, I will collect for the first time all of these variables to see whether
that's just what I've written down is when we talk about discrimination, these areas, I guess one of the problems with these big databases that have this wealth of longitudinal data is so many of the factors that you're wanting to look at, just there won't be the information there, right? And so that discrimination that sat there for so long, or just that, I guess all these factors is people just necessarily didn't think that was relevant at the time, or there were issues as to why they didn't ask those questions. So that's why they're not in the data sets. I mean, even the study that I'm in now, which has been going on for quite a few years, doesn't ask questions that I kind of think it should ask, because nobody thought to ask them at the time. But now it's only the last, because this is such a rapidly changing field of discovery that's the problem with some of this longitudinal data. So how hard is it to get I'm guessing you're having to start from scratch on some of this because it's just not there, right?
No, exactly. And to be fair, I've already published a couple of studies on this topic, but sometimes in databases, we don't even have information about sexual orientation. So we have to come up with some strategies to actually try and kind of identify these people. And a strategy that has been used by also other researchers is just looking at people that are in a relationship and comparing the gender or the sex of the participant of the study and of the partner, to actually compare people in same-sex and opposite-sex relationships. It could be a bias, because this population, we find reliably that older LGBT people, usually they're more likely to be single, so not to be in a relationship. So we're missing out a lot of these people. There are only a handful of studies that really looked into this issue. 3 years ago, when I had the first thought about this study, there were only 2. Now there are a dozen 3 years down the line. But I guess in general, they always approach this topic from a more of a social science point of view. I don't think, we haven't really kind of got into the details of looking at what happens in the brain and in cognition of these people. Also because in some of these databases, there aren't even many cognitive data to analyze. So I guess there are several limitations. So we need to actually recruit new people to test our hypothesis.
That isn't to say that these big databases that do exist are perfect either, does it? Because we know that they historically haven't necessarily made the right inroads to attract certain people from certain ethnicities or different groups, that they're not necessarily the best data sets in the first place. But I'd say it's only, I don't know if you would agree, it's only in my attendance at conferences the last couple of years that people are being called out when they present their results on the fact that there weren't any people, no Black people in these studies, or there were no Latinos in the studies, that certain populations of people were entirely missing from these trials. Yet we know that there are so many of those people where dementia is more prevalent for a whole range of reasons. So good work. So we're going to come back we only got 5 minutes left, and we feel we've only just started. So my first question was about MRI. So you all use MRI scans, and so MRIs are just old, aren't they? Right? I mean, it's old hat now. Why do we still use MRI in this old tech? And we moved on. It's all about pets now, and in about 2 sessions' time, we're going to have somebody who's talking about meg images and pet images. And what does MRI still have a place?
Absolutely, yeah, of course it does.
You're going to argue that because have you got one of these fancy, because aren't they scored by how many Teslas they've got? How many Teslas are in your MRI?
Well, so I'm not actively acquiring just now, but standardly in the NHS, you'll have like 1.5 and 3 sometimes. I've worked on 7, and when I worked with animals, I've worked on 9.4. So they vary. 7 is kind of generally research only for the most part.
Why animals work on a big one? Is that because you can't put a human in a 9 Tesla, 7 Tesla?
No.
Or is it just that we treat animals better than we do people?
So it's essentially, the more Tesla you get, the more expensive it is, and the more difficult it is to find a place to put it, essentially. But there's my physics maths will go out the window here, but there's an 11 point something. I want to say 11.6, but I'm probably very wrong. Sorry for anyone.
And the more Teslas you add, the more resolution you get.
Essentially, yeah. But there's an 11 point something going to Nottingham, I'm pretty sure.
So
fairly soon.
For people who are watching who don't know the difference, is this a bit like your cameras? You know, the first digital cameras had 2 megapixels, and now you can get a smartphone that's got 30.
Yeah, so like essentially take it back to the picture analogy that I had before. Yeah, you're gonna see your structure better as well. When I worked in metabolism, I could only see certain like aspects of metabolism at higher field strengths because, again, your resolution is better. So that's really the draw.
And can you? Does resolution make a difference to all of you? I mean, would you all want the best possible resolution? Would it make a significant difference? Is this the tech on the MRIs you're using a genuine barrier? Claire, you nodded, I think.
I mean, maybe if we could get slightly higher resolution. So I acquire images on a 3T scanner at the moment, and so I could do with slightly better resolution, just so I could get really into the kind of like smaller brain areas. But I could also do that if I just use different sequences. But then I wouldn't be able to get the whole brain. I'd only be kind of like zooming in. So
you can also get better resolution from zeroing in on a particular bit and doing it for longer.
I'm not a physicist, but just like tweaking your sequences.
Jodi understands this. I can tell. Jodi is.
Oh, I'm not a physicist either, but essentially, and without getting into the physics, essentially, yes. You want to acquire for longer. You want to acquire more slices of your image or things like that to try and get better resolution.
For anybody who's watching who's kind of interested in participating in a trial, is invited to have an MRI. I mean, I know that they're a bit scary, right? Because everything's super close and it's very noisy. But it just goes to show that even by having the scans done, you can contribute significantly to ongoing work now, both, whether you carry the ApoE gene or whether even research that I guess people who had this, who, looking at sexual minorities, wouldn't necessarily think of that MRIs could make a difference to that.
I think just on that point, I think it's probably sort of fairly important to say that you mentioned if MRI is old hat and things like that, and I know we're running out of time, but I just want to make the point that like MRI is the closest thing that we have to doing sort of dissection while somebody is still alive and with a technique that doesn't harm them. And the amount of information that we can get from that is monumental in various different ways, not just with what we're doing, but for various diseases and stuff. As a technique, it's saved like so many lives by the doors that it has opened. And the amount of information that you're giving a scientist if you're participating in an MRI scan for one of these sort of like trials or research activities or stuff like that is just huge. So it's definitely not done and out the door sort of thing.
Absolutely. Riccardo, just before we finish, is your trial recruiting right now? Are you looking for people? Is there anybody who's watching at home that's kind of not necessarily thought of themselves as somebody that would come forward in research? Is there a way that people can get involved?
I'm not recruiting yet, but I will at some point. So keep an eye on. But on the Brunel website, my study is advertised, so it can be seen. So definitely, it will be probably starting in kind of a year time. I guess in the first year, we will have to do all of the first retrospective studies.
Yeah.
But yeah, just very quickly, I just wanted to say that yes, we need MRI, even though we don't use 7 Tesla. What is important is the analysis that we do with the images and basically the questions that we want to answer with those images. That I guess is is still relevant to me. Yeah.
So all scanning techniques still entirely necessary. And Claire, I'm guessing your recruitment's finished now.
Possibly. So
you might need your next cohort.
Yeah, I've recruited 300 people so far.
Wow, well done.
Yeah, and I'm just sending off the latest batch for genetic analysis, but you never know how many of the E4s you're going to get in your sample. But I'm always looking for more volunteers. So yeah,
that's the issue. So particularly if you know you have E4, then by all means you'll find details on Riccardo, Jodi, and Claire and their Twitter feeds, and I'm sure they'll be tweeting when they're recruiting, because that's a big way to recruit on their webpage. So thank you very much. And to Alzheimer's Society that certainly fund your work. That's right, isn't it, Claire? And Alzheimer's Association with Riccardo. Jodi, how's your work funded? Do you get some charity money?
The Chief Scientist Office in Scotland.
Well, I'm sure you should definitely be applying to the next round of grants for the charities. Thank you very much, Riccardo Manca, Jodi Watt, Claire Lancaster, for joining us today. Thank you all.
Thank you.
Thank you.